today's webinar is all about using PDBe and PDBe-KB resources. So my name is David Armstrong, and my colleague James Tolchard is also here. He'll help me with answering some of the questions as we go along, and also helping us collate those to answer at the end of the webinar. Saves me having to worry about too many things at once. So this webinar is part of EMBL-EBI's Guide to series, and we've decided here to focus a bit on understanding binding sites in protein structures. Now, the way we're going to do this, I'll give you a short introduction to the resources we have, and then use a case study to walk through all those different resources that you can use to try and understand a bit more about important amino acids in ligand binding sites, and try and interpret those to use and understand for your future research. So to begin with, just an overview of the Protein Databank. So many of you will know that Protein Databank is an archive of 3D structures. Although it's the Protein Databank, it's a lot more than just proteins. There's the nucleic acid structures, DNA and RNA, but there's also structures with various different bound ligands, things like carbohydrates, but also drug molecules. And we can have a number of different types of entries with different proteins bound to different ligands. So there can be a vast number of different data that you can use to analyze structures. Now, the other thing to note is that it's an archive of experimentally determined structures. Now, the benefit of this is that you have experimental data to validate the structures against. So as well as validating on the model and the geometric plausibility of the structure, you have experimental validation to know how much you can rely on that data. Now, one of the things that really benefits us now in the use of PDBe is the sheer number of structures we have in the PDBe archive, and due to the growth predominantly over the last 20 years or so. And you'll see here that more recently, although the vast majority of structures in the PDBe are X-ray structures, more recently there's been a very small boom in EM, and I think this will keep going and keep increasing, and we're seeing a lot more electron microscopy structures in the PDBe. So we now have around 175 to 180,000 structures in the PDBe archive. So myself and James, we're from the Protein Databank in Europe, which is based at EMBL-EBI, just outside of Cambridge in the UK. We're actually part of a wider consortium called the Worldwide Protein Databank that together manage the PDBe archive and deposition of data to the PDBe. So some of you may be more familiar with the RCSB PDBe in the US. We collaborate closely with them and PDBe in Japan to ensure that the data that goes into the PDBe is as accurate and as consistent as possible to make it more beneficial for researchers. Now, we also have BMRB and the EMDB as part of this consortium, so that they help in terms of managing slightly different data archives, but together it means that we can link that data and ensure it's all consistent. So the benefit of having one big worldwide protein data bank is that we can have one system that we use between all of them that standardizes all these processes. And these processes allow us to get additional information in there in terms of identifying specific ligands, mapping individual sequences of proteins to UniProt, and also identifying linkages within the structure. We also have a standardized validation process as well. And all of these bits of information go into the PDBe archive. However, each of these sites have our own separate websites for presenting the data and sharing these with the research community. And during this process, we have slightly different processes to add additional annotations to the data, some of which will allow you to make different interpretations of the data and to use it in different ways. And so what I'm going to be describing today is the resources available at the PDBe and the way that we present that data to hopefully help you understand more about ligand binding sites. So one of the big benefits of doing all this additional linking data is we can provide more broad information about the science behind those entries, and it enables us to link biological and chemical information to what are inherently just a set of atomic coordinates in the PDBe archive file. So obviously some of this is done through deposition, but the rest of it we do after the data is, during the release of the data, to make it available on our website. So for instance, on the right here, we're turning something which is just individual atomic coordinates to something which becomes a bit more representative of what is going on with a ligand bound to a protein and an interacting residue shown. We do this in a lot more detail through our processes. One of the other benefits of PDBe is we are part of EMBL-EBI, and that means we have a whole host of other resources providing biological and chemical information that we can link to and we can work with to hopefully provide a more coherent explanation of the science that's going on in protein structures, and to provide you with more information through that as well. And one of these processes we have is something called Sifts. And what this process does is it integrates protein structure data at PDBe with protein sequence data at Uniprot. And it also brings in various other annotations from structure families such as SCOP and CAF, and also sequence-related annotations through Interpro, PFAM, and even gene ontology information. And we now link to data from Ensemble as well. So by tying all this information together, we can increase the amount of information that we can provide to give context to those protein structures. Now, one thing we've more recently done at PDBe is developing the Protein Databank in Europe Knowledge Base Consortium. So the idea of this consortium is to bring together all these different structural and functional annotations from distinct resources and processes, and to put them under one umbrella so that it's more easy to find all this different data relating to macromolecular structure. And we collaborate with a whole host of different resources to bring this information together. So this includes information on sequence domains, variation, drug binding, and lots more. And so this has been worked this on top of Sifts. Between the two of them, that really increases the amount of added value of that PDBe data. Now, in terms of chemistry information, we also have processes which enrich and improve the chemistry information that we provide for PDBe entries. So, for example, we add additional functional annotations for each ligand in PDBe. So where there are drug bank links available, or for example, cofactor annotation, we add these as well. And we also have embedded this into our search and our pages, so finding and understanding these annotations for ligands is much easier. And we've created a specific PDBe interactions software pipeline in order to calculate and define these interactions, and therefore be able to generate some interactive visualizations to be able to understand these interactions, such as the ones you can see on the right-hand side. And we combine this information with functional annotations from other PDBe-KB resources to hopefully give the most detailed information about small molecules and interaction data. So just in terms of what you might want to be aware of in protein-ligand interactions that are available, there are various different types of protein-ligand interactions. So we have covalent bonds, and although a covalent bond in theory involves this as creating one individual molecule, when it comes to PDBe data, we separate our protein molecules from even covalently bound ligands to simplify and clarify where the protein exists and where the additional bound molecules are. Otherwise, all the others are non-covalent interactions, such as salt bridges, hydrogen bonds. There are ring interactions, and also van der Waals forces and hydrophobic effects, which obviously drive protein folding, but also are very important in interactions of ligands with proteins. And so the graph here on the right is from a publication which analysed the interactions that are found within PDBe entries between proteins and a subset of small molecule ligands. And you see that actually there's a large number of hydrophobic interactions, although these are much weaker. But all these different interactions are found between proteins and ligands in the PDBe and may be important for binding of these ligands to the binding sites. The other thing to bear in mind when looking at ligands and binding sites is in terms of validation. And there's a few different points I wanted to make in terms of validation of ligands and helping you, when it comes down to making conclusions about ligand binding sites, make the most successful interpretations based on validation of that experimental data. And as I mentioned, this is why the PDBe having experimental data, and it's now mandatory for all techniques to provide that experimental data, and why that's important. So the first one of these is in terms of resolution. Now, as you'll probably be aware, the higher resolution you can get, the smaller number of angstroms, means the better quality of that data, the better clarity of the data for fitting those atoms into. So I just had a slide here to illustrate that. So at something like 3.7 angstrom resolution, you are able to fit large bulky ligands such as this heme within that density, but actually the accurate placement of atoms into that electron density data for a crystallography entry in this case is much more difficult. But as we increase the resolution, the accuracy of being able to position those atoms in the structure becomes much easier until you get to resolutions of around 1 angstrom, where you can really clarify the exact positions of each atom in the structure. Now, the other thing to bear in mind for X-ray structures is to use the difference density and look at the fit to the experimental data. So in examples where the model doesn't exactly fit the experimental data, we also show this red and green difference density. So the red density shows where there are atoms modeled, but there isn't actually enough experimental data to support those atoms being there. And the green density data shows areas where there is experimental data, but nothing has been modeled. So something has been missed. And in this particular case, it's quite a simple case. There's a water that we can get rid of, and we can flip around that tryptophan chain and much better satisfy the density data. Now, hopefully this is handled in most cases by the depositor, but we do need to provide this validation so that you can be aware of where there might be problems with the data and errors in the modeling. So in this webinar, I'm going to go through a particular case study just to highlight where you can get different parts of the information about ligand interactions with proteins through PDBe and PDBe-KB website. So the example here I'm going to look at is the inhibition of neuraminidase by Relenza, which is also known as zanamivir. And so this is actually an existing influenza drug that targets neuraminidase in influenza cells. So it's the viral neuraminidase that it specifically targets. However, it doesn't affect human targets, which is therefore beneficial as a drug. And the reason why it works is it outcompetes the natural reactant, which is sialic acid. And actually, if you look on the image at the top right here, the guanidino group circled in red here is actually in place of a hydroxyl in this case. So the one shown on the right here is zanamivir. And if you were to replace what's in the red circle with hydroxyl, you'd get that natural reactant of sialic acid. So in this kind of case study, I want to look at how we can analyze why this might be an effective inhibitor, looking at the interactions with the influenza neuraminidase protein, and comparing this with the interactions with the natural reactant sialic acid, and why this differs depending on the two. And then also to look at where we could go further if we wanted to understand more about designing new drugs or changing the way that we target this protein. So initially, I'm going to look at the PDBe-KB protein pages. Now, these are pages that we developed over the last couple of years, and they provide all the structural context for a particular given protein. So we do this by UniProt ID. So this means it's one specific protein from a specific organism, and it shows all the data based on the number of structures there are of that protein, how many ligands are found, and the interactions of those ligands with the protein, how many different protein interaction partners there are, and again, all the interactions between those. There's also a section for any additional functional annotations, particularly those that are developed through other PDBe-KB partners. And we also provide publication information as well, just to allow you to get a more rounded view of the protein. Okay, so let's start here at the summary page of the protein page here. So on this page, you get a bit of information about protein, and you also get these icons that highlight the different number of structures, ligands, and interactions in the structure, and also a component on the right-hand side here, which displays the best structure based on overall coverage and validation. And if you were to click on that, you can also view the structure in 3D. Now, in this webinar, we're going to be focusing on ligands and their interactions. So we want to go to the ligands section, and we can either scroll down the page or click specifically on the ligands icon. And we can see there already that there are 41 distinct ligands found bound to this viral strain of neuraminidase. So if you visit the ligands section of the protein pages, this gives us an overview of all the ligands and their binding residues of the protein in the PDB. So the first part shown at the top here is a list of all the ligands that are found bound to this protein in the PDB and how many they're found bound to. And it gives an image of the chemical structure of these ligands. So you can immediately see what these look like. You can also click on that image or the 3D view icon and view the binding site for a specific instance of that ligand bound to that protein structure. And now the second part of this section is something called a PropVista component. So PropVista was developed between ourselves, UniProt and Interpro, and it's a sequence-based viewer. But what we're able to do is we interpret the 3D structure data as a sequence-related view so that we can show all of the interactions between ligands and the protein based on that protein sequence. And when you minimize this and just show it overall, you can quickly begin to see the trend of which amino acids are important for binding to ligands for this particular protein. Now, if we want to look in a little bit more detail at the case that we want to look at, so in the case I'm looking at here, there are little crosses here where we can remove anything we're not interested in. So what I've done is I've removed everything except for SIA, which is sialic acid, and ZMR, which is zanamivir. So here you can see the comparison of the interaction pattern between sialic acid and zanamivir. And you can therefore make a comparison between these interaction profiles and see where they differ. Now, one thing that we can find quite quickly is that in this section here, where we have very similar interaction profiles, there is one additional interaction for zanamivir to this glutamate 229. We do also see a few additional interactions between sialic acid and zanamivir, but for now, we'll just concentrate on this one area where there are more interactions for zanamivir. So because we see this difference, we want to investigate this further and try and understand why this additional interaction could improve the strength of binding for zanamivir and allow it to outcompete sialic acid as a drug. So one other thing that we can do is compare all the other ligands and this interaction site. So this red arrow here shows that glutamate 229 that we spotted earlier for zanamivir. We found there are actually a few other ligands which also have this interaction. So again, if I wanted to look at other potential drug types or small molecule fragments that could be used for drugs, maybe I want to investigate these other ones as well. Now, one other thing that you can do as well when it comes to the ligand section of the protein page is you can download all this data as a CSV format as well. So if you want to do a more large-scale analysis, maybe something more programmatic, you can actually download all this data in CSV or JSON format by visiting, clicking on the format you request and then clicking download. Okay, so that's the ligand section of the protein page. I wanted to also highlight another section of the protein page, which is related to superposition. So we have created a process that for each of these protein pages, for each protein in the PDB, we superimpose all the copies of that protein in the PDB onto each other using a program called Gesamt. And it aligns these chains and clustered them based on their similarity. So we therefore can highlight where there are distinct structural clusters based on slightly differing structures in the PDB. And the other thing we could do is then also display the superposed ligand information as well, so that we can see where all the ligands bind. For example, on the right-hand side here, this is actually a protein from SARS-CoV-2 that causes COVID-19. And you can see where there's this area on the right-hand side where the vast majority of the ligands have been identified through PDB structures bind to this site. So you can use this superimposition to look a little bit more in a 3D view of where the important binding sites are on that particular protein. So if we go back to our neuraminidase structure, neuraminidase protein here, there's various buttons for viewing the structure clusters. We can do it with the view structure clusters here, which generally shows us the protein clusters, but doesn't show us ligands. But alternatively, we can click view all ligands, and this will show us where all the ligands are found within the PDB aligned to all these different clusters. So when we open this, we'll see something displayed a little bit like this. And in pink, just so that they really stand out, we highlight here all the different ligands and where they're found bound to that neuraminidase structure. So immediately we can highlight some key kind of binding sites for this protein. So obviously there's a large clustering of structures here. There is also a pattern of additional ligands on these areas as well. But for now, because we've already said we want to compare sialic acid and zanamivir, we're going to use this section on the right-hand side to hide everything except for those two ligands. And in this case, I've changed the colouring slightly so that the zanamivir ligand is shown with these grey carbons and other element colours, while the sialic acid is still shown in pink. So now we can more clearly see, we can see how similarly they bind to the protein, and we can see where this difference is between sialic acid, which has this hydroxyl here, and then zanamivir here, which has this guanidino group. And the other thing we can do is look at the different amino acids around, and it highlighted in pink here is that glutamate 229 that we spotted from the interactions earlier. And you can clearly see from here how it forms this interaction with this guanidino group. And obviously the distance there from the hydroxyl and the fact that it's opposite polarity in this case means that you wouldn't get that same interaction with sialic acid. So we can use this view to look a little bit more around the structure and generally get an idea of the difference between interactions between one amino acid and the other. The only issue with this view for doing that is that the protein structure that's displayed is only one representative structure. So you won't be able to see individual differences between specific ligands in that case. So in order to do that, we maybe want to look at a more specific example and interpret it in a bit more detail. So in this case, I'm going to go to PDBe entry 4MWX, which is a structure of neuraminidase bound to zanamivir. And here it is in the home page on the entry page for this. And if I click on that, it will take me to the ligands and environments page for that specific ligand in that entry. Now, what this does is it shows me a component in 2D, which highlights in detail all of the interactions. And this is interactive as well as a component. So we can hover over different regions of this and of the bonds and get information about what interactions there are actually occurring between the ligand and the protein, and in this case with other ligands as well, such as waters. And as we highlight things on here, these are also highlighted on the 3D view using our Molstar viewer as well, so that you can see this again interpreted in 3D. So I just wanted to show a comparison of the interaction component for 4MWX of zanamivir, and also with the sialic acid in a different entry in 5L18. Now, in general, the interactions between these are very similar, and around all of this region here, the interaction profiles are nearly identical. However, I'd just like to highlight a couple of points, which is firstly, the presence of this glutamate 229, which we identified before, and as you can see, forming an interaction with this nitrogen fat guanidino group. And the other thing is the presence of waters here in the sialic acid bound example, which are interacting with the hydroxyl, which is now left in zanamivir. Because of the change, because of this additional bulky group and the glutamate interaction, these waters have now disappeared and you don't get that interaction around here with waters. Although you get some of the similar water interactions around here, ignore the numbering on the waters because this is done randomly per entry. So we have to really kind of base this off similar interactions rather than specific water numbering. So if I look at that ZMR structure in a bit more detail, again, I can highlight glutamate 229, and this highlights it in the 3D view. So again, you can get a much clearer view of how this exists in 3D. So we've done quite a comprehensive analysis of comparing sialic acid and zanamivir and the difference in binding between the two. So the next question is where we might want to go next. So the example on the right here is the sialic acid bound structure. And so I've highlighted a few things here. We have the glutamate 229 here, and then that hydroxyl, which in the case of zanamivir is replaced by the guanidino group. And you can see here those two waters, which kind of take up that space between the two of these. And as we've shown, when zanamivir is bound, these waters don't interact, aren't found there anymore. And instead, this guanidino group kind of bridges this gap. So if we were looking to design another sialic acid derivative in order to develop new drugs, maybe we want to look at additional pockets of water and work out where we can add other side groups to a sialic acid to change that binding and perhaps create something which could be an even more beneficial drug. So in this case, I'm looking at a slightly different hydroxyl group. In the case of the left-hand image, it's this one over the other side. And you can see, again, there's a pocket of water nearby and a couple of other functional groups available, which potentially could interact more specifically with the protein. So maybe a change to this hydroxyl to something different would enable us to get a new drug target. And interestingly, in looking a bit more at the PDBe database, I was able to find something which is very closely related to zanamivir called laninamivir. And that has this hydroxyl group, although it's identical otherwise to zanamivir, it has this hydroxyl group instead changed to a methoxy group with this additional methyl on it. Now, obviously, this makes this a more hydrophobic interaction. And we did see on the previous image, this methyl group here, this hydrophobic group, perhaps it could interact somehow with that. Now, I haven't done any of more detail to look into this example, but I do know, having looked at Drug Bank, that it is actually in drug trials for treating influenza. So perhaps this could be a future flu treatment for us to use. Okay, so I'm going to leave the webinar there, just give you a summary of what we've talked about. So the key is that we now have such vast amounts of ligand and protein interaction data in the PDBe that allows us to do this large-scale analysis of ligand and protein interactions. And the PDBe-KB protein pages allow us to quickly identify these interactions thanks to the PropVista viewer and being able to really highlight those interactions on a large-scale level. And if we can compare these interactions, we can quickly see where there's similarities and differences in these interaction patterns to compare the interactions of these different ligands. And we also have the option to superimpose ligands onto the structures, and that can help you view the same data in 3D. And finally, if you want to look much more in depth at interactions of a specific entry, for specific ligands and a protein, then our interaction component really gives you a detailed view of this and allows you to make some conclusions about the important interactions and perhaps where you might target mutations of amino acids or instead perhaps designing new drugs. Okay, so that's all for the webinar today. If you do have any questions, please use the Q&A option. I see there's already a few questions gone in there, which is great. We'll try and answer as many of those as we can today. However, we won't necessarily be able to get through all of those. So if we don't get to your question, or if you have any other follow-up questions, we do have a help desk here that you can contact. And we also have all our various social media accounts. Again, if you follow us and if you want to contact us through those, again, to let us know. And finally, just before I finish up, is just to remind you about the remainder of the Guide to webinar series. Now, as well as these live webinars that you can register for using this link here, we also have all of the recorded webinars available as well. So you can go back and look at all those and use this nice new EBI training pages to find other training resources related to PDBe services and also others from EBI. And finally, just a reminder to fill in the survey that launches when the webinar is finished. We'll be really grateful if you can give your feedback and provide us with some ideas about what you might want to hear about next time. So that's where I'll finish, but we will come back shortly for a Q&A. We'll just collate some of those questions between us, and we'll be back shortly.
Okay, David, shall I start at the top of the list and we can have a look through?
Yep, that sounds good.
Okay, so first question here is, what is the criteria for ligand binding sites? Is it just by distance? And if so, what is the cutoff?
Yes, so we use a process through software called RDKit to calculate interactions between ligands and proteins. So it does use a distance cutoff. However, it uses various different distance cutoffs because it's not just looking at only certain types of interactions. It's actually looking at ionic interactions, hydrogen bonding, plane-plane interactions, and obviously that distance cutoff is different. So it uses a knowledge of the types of atoms involved in those interactions, and applies then a cutoff specifically to those. So if you want to know more about that, actually underneath the interactions component on our page, there's a little short summary and a link to find out some more information about how we do that cutoff. But we use that same data there in those interactions as we do in the PDBe-KB pages as well.
So we've just had a quick question that I know I can answer very quickly. Somebody's asked about if it's possible to get a certificate for this training. So unfortunately, we don't actually provide certificates of attendance for our webinars. So I can just confirm that now. So shall we move on to the next question, David? So somebody's asked, what if we come across a protein which does not have a 3D structure? Do you have or know of some tool to predict it from the amino acid FASTA sequence?
Yes, so in cases where there is no protein in the PDBe, there's kind of two options of going for here. One option is to actually look for proteins with significant homology that do exist in the PDBe. And actually, if you get quite a high sequence homology of, say, at least 30% sequence homology, you can be reasonably confident the structures should be quite similar as well. If there is no homologous structure available at all either, then you probably want to use some sort of structure prediction tool, of which there are various. There are services such as Swiss Model, and there's also servers like ITASA or Fire2, which can provide you with structural models based on that. And there's, I mean, one of the things, the big news in that field recently is the addition of the DeepMind's AlphaFold server, which is, or not server, it's a software which they have used to try and improve the structure prediction. At the moment, those aren't available for you to publicly use, but there are various services that are through the same competition that Google was shown that the AlphaFold service was shown to be very good at. There's a service called CASP, which is used as an assessment of how good these protein structure prediction services are. So if you want to look at perhaps which one is scoring best in CASP that you can then access, that is also another option.
Okay, so next question. So concerning epitope mapping for antibodies, different methods such as PET scan, NMR, and X-ray crystallography provide quite different results. Is there one gold standard technique outcompeting the others, or is a subsequent alanine scan obligatory to define the key interacting amino acids?
So I think I probably can't give you a very clear answer on it, the exact information relating to antibodies. I mean, one of the problems we have in terms of determining interactions between antibodies and proteins is that those interactions are so specific that you don't necessarily, you're not able to get the same kind of interaction pattern and predict it as well as you might do for other protein-protein interactions. Yeah, however, in that particular example, unfortunately, I'm not clear enough on the area to be able to give you any more kind of advice of which process is best to use. I'm not sure whether James has any more suggestions on that at all.
No, that's all I've got, Dave.
Okay, great, thank you.
Okay, so as a very quick question, I think. Is there a colourblind friendly mode within PDBe?
There isn't a different mode. When we design our services, we do try to design them with different colour blindness requirements in mind. I know one of our developers himself is colourblind, and we do try and take that into account. If there is something that you are finding difficult, then it's worth letting us know, and we can look again at our colour palettes that we use and try and adapt these to perhaps be a bit more clear. But in terms of unless you have a browser which I think you can have browser add-ons which do it automatically for you. We don't actually have a service ourselves to do that.
Thank you, David. So next one, is it possible to look at the predicted interactions of a protein when it has post-translational modifications?
So this is an interesting question, because we have various protein structures, some of which may have be modeled with post-translational modifications and some of which may not. Some of the PDBe-KB resources, some of the partners through the PDBe-KB resource do provide data related to post-translational modifications, and that might help in terms of identifying some of these examples and using it to compare with the interaction data. But obviously, in terms of what's in the PDB, it's not clear whether there should or shouldn't be post-translational modifications in each case, so we don't really handle that ourselves. But yeah, using some of those additional PDBe-KB resources and then cross-referencing against interactions and looking at, in the structures section, it does say if a residue is modified, if it is modified in the structure. So you can also cross-reference that as well and use that to help kind of determine the importance of these sites.
Okay, thank you, David. So James has provided me another question here in our chat. Is there an API for me to turn my protein ligand analysis into scripts or a notebook?
Yes, so we have, so actually all the data on PDBe's web pages is served by APIs. So we have two APIs. One is an entry-based API, which provides all the data specific to an entry. And that includes ligand-specific information as well. However, we use, for the protein pages, we use a graph-based API. So we call that an aggregated API because the data it provides is aggregated per the protein you're looking at. And it's that API which provides the underlying data, which we show in the interaction section. So one thing I would recommend that you look for is on PDBe's training pages, we have a link to a webinar series that we run last year. And that was all related to different ways of accessing data at PDBe using programmatic access. And that highlights the different APIs we use and how to use those. So I'd recommend you look for those and use those as a way to try and understand a bit more about using the API to access that data.
Okay, thank you. So next question from James. Is there a way to filter out all structures binding to a particular ligand and download all of them at once?
Not currently. However, that is actually something we are working on at the moment. And so we hope to develop a very similar resource as the protein pages, but instead for ligand pages instead, with the idea as it would show all the data related to a ligand and allow you to see all the entries it's in and all the similar ligands. However, that's currently still in development. It's also part of a collaboration between our colleagues at Kemble and also at the CCDC. So we're still developing that, but we hope that we'll soon have those pages available, and that will allow you then to do that. The other way to approach it, if you are programmatically, you're able to do it programmatically, is that you can use the API to get a list of all the PDBe ID codes for a particular ligand ID, and you can then feed that into a download surface we have, which you'll find linked from our pages, and that download service allows you to then bulk download all of those entries. So that is another way to do it, at least in a crude way, but hopefully we'll have a much better way to do that sometime, maybe later this year or next year.
Great. Our next question on the list, is there a tunable threshold for what counts as interaction, or another way to tell a strong interaction from a weak one? Yes,
so because we use RDKit, we just use their thresholds, the define thresholds through there for what defines each type of interaction. Really what tells you the difference between a strong and a weak one in general is the bond distance. The closer two atoms are, in theory, the stronger the bond is, but also the interaction type. For example, an ionic interaction, and when you go on that interaction component and you hover over those dashed lines, it will tell you the type of interaction that those two things are forming based on our classification through RDKit. So, for example, an ionic interaction will be stronger than a hydrogen bond, which would be stronger than a van der Waals interaction. So it'd be worth looking at those and maybe cross-referencing that against those different interaction types and the expected strength of those bonds for each interaction type.
Great. James, did you want to come in?
With particular questions. So I'm going through trying to answer them in text as well. Let's have a look. So is it possible to determine the binding affinity of different ligands with proteins?
So that isn't something that we calculate at PDBe. We do link out for ligand pages, for our ligand environments pages, we do link out to other ligand services such as EMBL, who do provide information on statistics relating to ligands, which can include things like that. So yeah, it's not something we provide ourselves, but hopefully through some of the links out, you can get that information from elsewhere.
Yeah, I'd say it's very difficult to assess that computationally, but you can try to approach it at least qualitatively with metrics such as binding energies. FoldX, for example, can provide.
So we have another question here. Can you study model ligand interaction with protein where ligands are peptides and you do not have a PDBe entry?
So, well, if you did have a PDBe entry, then actually peptides would come under the macromolecular interactions section on the protein page. However, in the case where you don't have a PDBe entry, yes, you can model these, but not through any PDBe resources. I think probably the most used software to do this is software called Haddock, which can be used to model interaction of ligands and other molecules with proteins. I don't know if you have any other examples, James, that might be used to do this.
Autodoc is what I would have used.
Okay.
Okay, so we've got a few minutes left, I think. So let's try and get through some more of these. We have, can we run the ligand interactions analysis and superposition for a protein ligand system modeled through computational molecular docking?
Okay, let me just untangle that one. Okay, so the ligands interactions pipeline, we do actually make publicly available on our GitHub account. So you can run that interactions analysis pipeline separately. Currently, our superposition process we haven't made available publicly yet. It also uses external software called Gazamt. However, that's something that maybe we might need to look at in terms of can we make those. We are trying to constantly trying to make more of our processes and software available open access for other people to use. That's probably our newest kind of service that we've started. So at the moment, that's not available open access. But the ligands interaction pipeline you can use with your own data by adapting the scripts that we use on GitHub.
Great. Can we study how different environments affect the interaction on the web page?
I don't think you could do that directly. It very much depends on which environments you're looking to vary and understand. In cases where within a subset of structures and another subset of structures, they are in a different environment, or they have maybe a different other ligand bound or a cofactor bound within the structure, you could then perhaps compare those in a more manual process. But it's not something you could do directly through the page. And I think if you're looking at something like this, it might be better to look at these through an individual entry-based view. Try and pick up some structures that you know from publications have been analysing these different environments, and then compare those interactions from there. But it's something that would require a little bit more workaround than just necessary accessing PDBe resources.
Okay, thank you, David. So next question we have, what are the most common challenges faced in identifying the binding sites on proteins?
Okay, well, I suppose one of the big challenges in terms of using PDBe data is actually ensuring that the data that is going into that analysis is accurate. And that's why we provide validation reports, so that hopefully the depositors, when they deposit the data, are depositing as accurate as possible structures, in order to ensure that the binding sites hopefully are then also accurate. I guess one of the other things is limitations of how many structures are available. So in the cases we've shown here, we had 50 odd structures with around 40 ligands. And so it's much easier to identify the binding sites because you've got that redundancy of structures with different ligands bound. And so you can more confidently say that those are important interactions. Whereas if you have a smaller number of different ligands bound, then you'd probably have to do that more at an entry by entry basis, look more closely at the validation, and do it in that way.
Okay, thank you. So James is typing on that one. So possibly a quick one here. Is there a rule of thumb of what distance is considered an interaction for different types, e.g. salt bridge, hydrogen bond, aromatic stack?
Yes, although it's dependent on the atom types as well. So obviously interactions between certain atom types will be slightly different length bonds or slightly different lengths of interactions than for other atom types. Off the top of my head, I'm not going to try and reel off numbers because I'll just give you incorrect ones. But I'm sure if you look those up, there should be some resources which give you kind of for different atom types, the expected hydrogen bonding length for those interactions and that kind of thing.
Okay,
so just to add, actually, in terms of ionic interactions, that could in fact be affected by pH as well, because the strength of those charges might be different and therefore might affect the linking as well.
There's a question here asks, do you have any data in the PDBe about protein-protein interactions? So, I mean, there's two parts to that, because I think we can probably direct you to the right resource, but do you want to answer on behalf of the PDBe part there, David?
Yes, so the section below the ligand section on the PDBe-KB pages is dedicated to protein-protein interactions within PDBe entries. So you can use that for understanding interactions between proteins in the PDBe. However, as Tom's alluded to there, we also have other resources at EBI. So if you were to go to Complex Portal and Intact, they look a lot more at the interactions between proteins and use the use data in publications and curate that data to understand more about those interactions. So potentially you can get some of it through the PDBe, and there's also a range of that data also available through those resources as well.
Fantastic. So let's see the next one. Maybe have one quick question, and then I think we should probably move to wrap up. So question here, is there a way of predicting or compensating for the movement of the protein structure in vivo rather than the rigid crystal form?
That's a very good question. And in terms of the interaction data that we display, we don't do that. I think you would need to look more individually at particular structures. You can, for example, for a crystal structure, you could look at the B factors of specific atoms in the structure, and that might highlight where there are more significant motions in the protein chain. But you can also use NMR entries as well, which give the range of different conformations that the protein might take up in solution. And again, that will give you an idea of where there is motion in the protein. In terms of predicting from those different motions or different mobility, how that would affect interactions, I'm not sure you would be able to do that unless you were to use a software that models it for you. So for instance, using different conformations and using for maybe Autodoc, like James mentioned before, or Haddock, for example, use a range of conformations and then model that from there, unless those different conformations already existed with the ligand bound in the PDBe, and then you compare those structures.
Okay, fantastic. So there was also a question here just about providing links to resources. So we'll be making sure that we have access to the slides that David has presented afterwards. And of course, this recording will be available. So if you do want to go back over and listen again, you should be able to find out the names of those quite easily. And hopefully they're things that could be found quite easily either through the PDBe website or through a quick internet search. But I'm also very conscious of the time that we have now. So I think, David, if you're happy, should we wrap up here?
Yeah, that's fine. Yeah, thank you very much.
So thank you very much to all of our attendees who are still with us. It was great to have you joining us. Obviously, we'll let you know in a couple of days when this recording is available, as the slides there as well. And I encourage you to obviously contact the PDBe if you have other questions. I know there's been a lot that we've managed to get through, but there are still many that we haven't been able to answer. So sorry, we weren't able to get through all of those. And please do check out some of the other webinars that we'll be running in the future. So we'll be taking a short break over the summer here in the UK, but from September onwards as well, we'll have a full program of other webinars as well coming up this autumn. Okay, so thank you very much, everybody, and goodbye, and hopefully see you all again soon.
Thank you, everyone. Bye-bye.